On to translation, second half of gene expression. Transcription was pretty basic, right? And the term made sense. You're going from a nucleic acid language, nucleic acid just like a medical transcriptionist just writes what's said. But in translation, we're moving to a new language, a protein language, just like translating from one language to another. But this one, the big trick is, RNA is written in a code of 4 different nucleotides, but now we're moving to a protein language with 20 different amino acids. So we can't just have A stand for methionine. We're going to have to do a little more than that to get this done. Plus there's just more players involved. Well, let's dive into it. Just like with DNA replication and transcription, translation has the same 3 stages. We're going to start, keep going, and then stop. But to get started, we need to have everybody at the party. So let's look at who's supposed to show up to get translation underway. Well, starting with the obvious, messenger RNA, which was produced via transcription, is here. We need to get that to a ribosome and fit that messenger RNA between these 2 subunits, kind of thread it through there. We also need tRNA. This is also made by transcription. And it's a complicated, twisted bunch of RNA, but a lot of times we just sort of shortcut it with this little cartoon. And it carries with it amino acids. So remember, amino acids have this common structure. They varied based on their R group. And we'll usually just symbolize those with these little shapes. And then we also have GTP, cousin of ATP, which is going to provide the energy to string these amino acids together. Well, RNA is the star of the show here, so let's get to know messenger RNA a little bit better, and then tRNA. So messenger RNA is written in 3-letter sequences known as codons, and there are 64 of them, which you can see on this chart. Kind of a boring chart, but this was the result of years of research by many scientists, so it's really quite a remarkable production. And this tells us which amino acids each codon carries, plus a stop codon. So each of these 3-letter abbreviations, this one stands for leucine, a specific amino acid. And then there's also the stop codon. Obviously that's going to come in with the termination side of things. And so let's look at the messenger RNA molecule right here. We have this first string of nucleotides, UGG, and we can look over at this codon chart to find UGG. It's right here. And that's going to code for the amino acid tryptophan. So tryptophan goes with that one. But how do we get from one to the other? We can look it up on this chart, but there's no little person inside of your cells that can look it up and say, hey, we need a tryptophan over here. Instead, they use transfer RNA, or tRNA. That's the key to the genetic code. It takes us from the nucleic acid language to a protein language. Let's say, for example, we had on the messenger RNA the code UUC. And if we went and looked that up, let's go ahead and do that. What is it going to call for? We look it up on this chart. UUC codes for phenylalanine. All right, so we know we need that. But again, the cell can't do that. So its go-between is the tRNA. And this simplified version highlights the anticodon and the amino acid. Notice this anticodon is complementary to the codon. So AAG, all of those are complementary, and it carries with it the amino acid phenylalanine. So it physically is attached to this tRNA molecule. And that's the key to the genetic code. That's how we get translation done. Well, let's look at the whole process in play. All right, so let's get started and initiate translation. Here's our messenger RNA molecule. We got to put a ribosome on there, but we can't start at the very first nucleotide. We need to move in a little bit, but where exactly? Let's take a look. So in initiation, we have our ribosome and we have our messenger RNA. But the question is, where do we start? We need to establish the right reading frame. And as we can see up here, we always start at the start codon, which is always AUG, and the tRNA that corresponds to that brings in the amino acid methionine. Once we get started there, then we can spend some energy, GTP, bring in the rest of the ribosome, and we're ready to go. Well, why is it so important to start just there? Well, take this sentence down at the bottom of the screen. A little hard to read, but it's all written in three-letter words, and if you put spaces between them, the dog ate one cat. Okay, a little gruesome, but there it is. What if you started at the second letter instead of the first, but could only read three-letter words? That would be a little bit like starting at the wrong spot on the messenger RNA, and it would look like this. Het ogateo nek et. I don't even know what language that is, but certainly if you kind of move that over to the DNA protein realm, it is not going to give you the right protein. So it's important to start at the right spot. So then we move on to elongation, this repeating process. So it's just shown as a cycle. It keeps just doing the same thing. So let's pick up where we have the whole ribosome assembled. We have this tRNA with a long polypeptide strand, so each of the amino acids is symbolized by these little purple shapes. And at this site, we have another codon that this tRNA matches with its anticodon, right? They're complementary, and it carries another amino acid. So once that's recognized, we spend some energy in the form of GTP. And with these two tRNAs sitting right next to each other, then a bond can form between these two. And so here we see that the bond has formed, and this whole polypeptide chain has been transferred onto the tRNA in the A site right here. Well, we're going to spend a little more energy, kick the old tRNA out, this empty. It's going to go off and find another amino acid and get recharged, and then it can come back and start helping with more translation. And the other one shifted into that P site. And then the process repeats. And that's elongation. Basically, complementary base pairing between codons and anticodons and the forming of new peptide bonds between amino acids. Let's finish up translation with termination. And that's when the ribosome reaches one of these three stop codons on the messenger RNA, which brings in a different type of tRNA. It's a release factor. There's no amino acid carried with that one. It just signals for all of the rest of these to start falling apart. We have a free polypeptide that can go off and do its job somewhere, a ribosome, large and small subunits in the messenger RNA. They can go off and make more proteins themselves. But that's how the process is finished. All right, translation. Done, done, and done. And now we have a protein. Well, really, it's just a string of amino acids at this point, but remember that the shape of a protein was crucial to its function. So the next thing we need to do is fold these up. So far we have strung together these amino acids. And if you remember the basic structure of an amino acid, this R group was the one that was really different. And the interactions between an R group of one amino acid and another one created these different spirals and sheets and bends and folds and twists to give it that three-dimensional shape. So that's the next thing that happens to the protein, and a lot of that's done within different organelles. Where does that protein go next? There's a couple places it could go, but let's do a bit of a recap. Here we had DNA in the nucleus, and we did transcription to get RNA. So there we have RNA. It needs to then leave the nucleus and go to a ribosome. So at that ribosome, we do translation and crank out a protein. And ribosomes come in a couple different forms. Those that are free, they're not associated with anything else. They're just floating out here in the cytoplasm, here and here and here. They make proteins that usually stay within the cell. Things like the cytoskeleton, enzymes, etc. But if we need to get that protein out of the cell, let's say it's a hormone or a digestive enzyme, then we need a different system. We need kind of a routing system. We need something a little more like UPS or FedEx or the post office. And that's going to involve these other organelles that we refer to as the endomembrane system. So let's take a look at that one. So we had our RNA here in the nucleus, and that's going to leave, and it's going to go through a series of other organelles. So this brown one here next, it's going to go there, and then to this green one, and then it's going to leave the cell. Notice how this brown series of membranes is connected directly, you can see this little pore right here, to the nucleus, so that RNA inside the nucleus can leave and come directly into here. And it is called rough endoplasmic reticulum because it has all these ribosomes that give it sort of a roughened appearance under the microscope. And so that's where the proteins are produced. But instead of floating off free into the cytoplasm, they stay within the rough endoplasmic reticulum. And then several of them are grouped up and shipped out in a secretory vesicle. So a vesicle is just a pinched off piece of the membranes from the endoplasmic reticulum. So here we have that one here filled with proteins, and then it goes to the Golgi apparatus. And that's kind of like the post office. It puts some chemical stamps on it and puts it in the right bin so that it can be shipped off to the right city. In other words, it does some processing. It groups the right proteins together and it tells them where to go. And once that process is done, then it's going to pinch off another vesicle full of those proteins that then merges with the membrane itself. Since this secretory vesicle is full of lots of proteins, when it merges, then all those proteins spill out into the extracellular fluid. And that's done by a process known as exocytosis, which you'll learn about next. Well, there is translation. We've gotten from a coded message written in a nucleic acid language to a string of amino acids that we folded up and even shipped out of the cell. Remember that all this happens at the cytoplasm, at a ribosome, and we use codons of messenger RNA translated by anticodons on transfer RNA to help us string together amino acids. And that together is gene expression. Now that we have a protein, we can do something with it, whether that's produce pigment in our hair, or an enzyme for the stomach, or a hormone that's going to be sent out to control what the body does. The proteins are really where it's at. They're crucial to how life functions, and it's important that they get made just right. I again remind you that there are plenty of good animations. Once you have the basics down, that will really help bring it to life and help you appreciate this truly remarkable process.